So thank you very much for the invitation, the chance to come to this meeting. I'm looking forward to all of it, and then hopefully try to kick you off in the right direction. I always want to start. Yeah. Well, that's probably the best way to experience my talks without actually hearing them. So they're actually more entertaining if you don't if you just watch my hand signals, right? Okay. So sorry about that. Is that okay? Can everybody hear me in the back now? Right? We're okay. Okay, good. Be sure to yell at me if I get too far away from this microphone. So anyway, thank you all for the opportunity to come and talk to you today and talk to you a little bit about how we view some of the issues that go with hyperphagia associated with what happens in obesity. And I think the idea of trying to understand this as it relates both to the problems that are associated with obesity, but also problems that are associated with other kinds of syndromes, I think are extremely important and a fruitful way to try to understand the system. I will start with disclosures just to make sure that we're all on the same page and so that Senator Chuck Grassley doesn't come looking for me later. I work with a lot of different companies that are interested in bringing treatments for obesity and diabetes to the marketplace. I think what you'll see today is I'm talking about rosiglitazone and other TZD drugs, and I don't have any relationship with companies that work on them. All right, let me start with something that all of us know, right? The obesity epidemic gets worse. We don't really know why it's getting worse, right? Trying to identify the causes that are changing in the environment to be able to say why it is that we're worse now than we were 30 years ago is no trivial task. But I think everybody understands, at least at some level, that what we eat has an impact on what we weigh, right? Now, with that, there's lots of confusion, right, about what it is that we're eating that makes things worse, right? If you read the popular press at all around this, it would be very easy for me to become confused. I think it's extremely easy for the lay public to be confused about what it is that is driving. And again, it is complicated, but I'm going to show you animal data that allows you to at least address one issue and be more clear about that. So this is two groups of mice, right? One is maintained on a low-fat diet, and it's a very low-fat diet, a low-fat diet that any vegan would be proud of, right, at about 6%. And the other diet is a group of animals that are maintained on a high-fat diet. That high-fat diet happens to be 40% fat, with most of that fat coming in the form of butter oil, right, so saturated fats. And we built it to look like what a diet looks like, at least in Cincinnati, Ohio, okay? And what you'll notice is these diets are very well matched, right? And despite the fact that they're very well matched, what you see is a different in weight outcome, right? The animals that are maintained on the high-fat diet that you see in the top line, right? I don't know how well you can see that, right? Have increased weight. Why do they have increased weight? Because they eat more, right? That's why they gain most of the weight, right? So again, we get into all sorts of issues about what the biochemical differences in terms of the calorie load and how the calories are being utilized, that gets a lot of conversation that's appropriate. But the fundamental thing that happens that differs between the two diets is that the mice that are exposed to the high-fat diet consume more of it, and that drives the majority of them, right? They are hyperphagic, at least for some period of time. And so a lot of what we do is try to understand, well, how that happens, right? What is it that drives increased consumption of that diet? So the question is, how do these high-fat diets contribute to obesity? Part of the answer is that what we eat has an impact on the key signals that regulate our food intake and body weight. And I'll just give you this one example, right? So this is an experiment in which rats are treated with a hormone called leptin. Most of you are familiar with it. It's a hormone made in adipose tissue that reflects how much adipose tissue you have. And it's a key component about why it's hard to lose weight, right? When leptin levels fall, right, you engage a complex neuroendocrine circuit that makes you burn less calories and makes you hungry, right? And what's interesting is what's sensible here is that when you give animals more leptin, that is, you reflect to the animal that it has increased levels of body fat, the animal does something sensible in reply and says, well, I don't have to eat as much then, right, in order to keep my the amount of body fat constant. But what's interesting is you take another group of animals and give them a high-fat diet and give them exactly the same dose of leptin, you can see that they're entirely resistant to that effect of leptin. And that's true whether you look at their food intake or the weight loss that comes with leptin treatment. So the point about this is that what we eat has a distinct impact on the ability of the leptin signal to get to our brains and provide the appropriate feedback about ourselves. Right? So again, what we eat has an impact on the signal, right, that matters. So this is a review that we wrote recently that appeared in Cell Metabolism that I would encourage some of you to read. The idea here is a very simple one, right? That the difference as you go from the lean state to the diet-induced obese state really has to do with the threshold for how much leptin you need to prevent it from engaging in responses in the CNS that would, again, push you towards eating more and burning less. And leptin resistance, the inability for you to respond to leptin that's caused by the diet, is a key driver for why it is that you defend a lower weight when you're on a low-fat diet versus when the animals are on a high-fat diet. And again, the same mechanisms are involved whether you're defending it down here or you're defending it here. Right? It's the same set of systems. The question is the threshold by which you have to have how much leptin is necessary in order to keep the system working the appropriate way. Okay, so one of the key questions then from our perspective is, what are the molecular underpinnings that make it so that translate what you eat into a reduced ability of the hormone leptin to drive a change in food intake or body weight? Okay? So this very talented woman who has worked with me over the last several years by the name of Karen Ryan. And Karen got interested in the idea that part of what's driving animals from here to here has to do with activation of a receptor called PPAR gamma. Okay? So what's PPAR gamma? PPAR gamma is a nuclear receptor, transcription factor, that is turned on by different kinds of nutrients. Right? So it controls a number of components of target gene transcription, including those that are involved in adipogenesis, lipid, and glucose metabolism. But what is it at its core, right? Well, again, what drives the endogenous activity of PPAR gamma? Well, our best guesses are that it has to do with fatty acids and fat metabolites that drive and activate the PPAR gamma system. Now, why is this interesting? Well, the reason it's interesting from a clinical perspective is that drugs that activate PPAR gamma have been used until relatively recently to treat diabetes, right? But the real role for PPAR gamma is to act as a general sensor of the flux of fatty acids. That is, these drugs that we built were not built, Mother Nature didn't give us PPAR gamma to respond to GlaxoSmithKline's drug, right? It was here to respond to fatty acids. So again, the idea here is in the case of TZDs that they can bind to the same pockets and they drive supraphysiological activation of this system that again results in adipogenesis and a control of glucose metabolism. Now, the problem is that along with improved glucose levels comes increased body weight. All right? So again, this has obviously always been an issue of taking folks who are likely to potentially have weight issues from the beginning, have glucose regulation problems, and what we do is we give them a drug that helps their glucose, but actually makes the weight worse. Right? And that's what you see here, right? So in both humans and in rats, it's very clear that rosiglitazone, right, a PPAR gamma agonist, can cause increased weight in both of these situations. Right? Now, here's the interesting part why I'm talking to you at a hyperphagia meeting about this. If you went and talked to 90% of the endocrinologists who would prescribe TZD drugs and said, why do the patients gain weight? They would say it's because you had adipogenesis, right? You put on more fat, right? More fat cells got born. Now, I'm a neuroscientist by training, and I can tell you that explanation made zero sense to me, right? Because if you don't change something about the energy balance equation, it doesn't matter how many adipocytes you have. You're not putting more or less nutrients in them. So you have to be able to change one side of this equation or the other in order to have an effect. And again, our hypothesis was that this involved the central nervous system. Here's the first bit of data that said, boy, this really does have to involve something we hadn't thought about. So this is peripheral rosiglitazone given to rats, and you look just in the first 24 hours. And what you see is a small but clear effect for the animals to be hyperphagic in response to TZD drugs. I bore you with that small fact because in the first 24 hours, there's no new adipocytes, right? It can't be that what we're explaining here has to do with the production of new adipose tissue, right? It has to do with the fact that what's going on here is that the animal is eating more, okay? So how does that occur, right? So again, the notion about how PPAR gamma acts is that you find it in macrophages and adipose adipocytes in the periphery, and then that's what drives the benefits from TZD drugs and the side effects, right? But what turns out is the case is that we had hypothesis that, again, for it to happen in 24 hours, it had to be the case that something's going on directly in the central nervous system as a result of these drugs. Okay, so into that came work from Mike Schwartz's group at the University of Washington, in which they found that there is very clear evidence of PPAR gamma distribution in a number of key regions in the hypothalamus that we know are important for the regulation of food intake and in fact are important for the regulation of leptin action. So again, this supported the idea that maybe PPAR gamma was important in the brain. Also here is in the paraventricular nerve. So how do you go about testing that? So here's what Karen did. What she did was to take animals and give teeny tiny doses of rosiglitazone, but to give them directly into the central nervous system to turn on PPAR gamma. And the prediction was that that by itself, without turning on PPAR gamma in the periphery, would cause increased food intake, body weight, and body fat. So here's what you see, right? So here's a small dose effect curve for, again, central administration of rosiglitazone. And what you see is that after this tiny dose of rosiglitazone, the animals consume 50% more calories in the first 24 hours. Right? Again, pretty profound response to tickling this one receptor. It gets worse, right? If you just do one administration and you look over several days, the animals go on increasing their food intake for 3 full days. If you come back 7 days later and take a look at the animal's body fat, what you can see is that the animals who got this small dose of rosiglitazone had an increase in fat mass that occurred over the next week. So the point here is that just tickling that one receptor directly in the central nervous system has a profound impact to cause increased food intake and weight gain. Now, for a variety of reasons, what you'd hypothesize is that turning on PPAR gamma should look like what happens on a high-fat diet, right? So what you need is a chronic administration paradigm. Now, for a variety of reasons, doing this purely pharmacologically just wasn't an option. So what we did was to take a trick out of the PPAR gamma literature, particularly from an investigator at Penn by the name of Mitch Lazar, who had made a PPAR gamma version that was constitutively activated by connecting it to this viral protein. Okay? And then we put it into a vector and now administer that into the central nervous system to add constitutively active PPAR gamma receptors into these key circuits in the hypothalamus. And the result is, again, you get this more than doubling of fat gain over the next several weeks. So again, turning this on now chronically has an effect both on food intake and body weight over periods of time. Okay. Now the question is, well, this may be true, right, that turning on PPAR gamma in the brain matters, but is it true that that's relevant to why TZD drugs actually drive increased weight? Right? So the experiment here is we're going to give rosiglitazone in the periphery, but what we're going to do is prevent PPAR gamma activity in the brain by a variety of methods and see whether you still get the same ability to drive increased food intake by peripheral rosiglitazone. Okay, so here's the experiment. So here what I showed you before, right, is that rosiglitazone shows you this nice increase in food intake in the first 24 hours when it's given peripherally. But if you now give an antagonist in the brain to prevent it from acting on PPAR gamma in the brain, you can completely eliminate that response. Now again, there's always issues about pharmacology here, so we've also done this with a viral approach, right, where we would knock down PPAR gamma receptors using shRNA. And here it is in the control condition. You can see the effect of rosiglitazone. Oops. But when in the condition where we've knocked down PPAR gamma receptors, that response is absent. So again, the point here is that peripheral rosiglitazone treatment that leads to weight gain has an important component that happens in the central nervous system. Now, this paper appeared last year, and it appeared with a companion paper from Jerry Olewski's group in which they had knocked down PPAR gamma in the brain using a Krelok strategy. And they showed not only, again, that peripheral rosiglitazone was less effective to cause weight gain, but also was less effective to improve glucose regulation. So that even some of the glucose benefits of rosiglitazone probably have a relationship to activating the CNS system. Okay. So now I've shown you two experiments, right, where we turn on PPAR gamma in a variety of ways, either by constitutively activating it, giving it in the central nervous system, or giving peripheral rosiglitazone, and showing you that it can matter. But does that mean that, in fact, that PPAR gamma receptors matter for normal regulation of food intake and body weight? After all, that could be a parlor trick, right? After I've shown you a bunch of situations that are artificial, right? That we have done something to turn on PPAR gamma in a way that has little to do with normal physiology. So what you really have to do is not these gain of function experiments, but loss of function experiments, right? Can I block the system in some way and show that, in fact, I can reduce food intake? So the first set of experiments, Karen uses an antagonist to PPAR gamma, gives it in small doses into the central nervous system, and again, the expectation is we should see reductions in food intake and body weight. And here's, in fact, what happens, right? So here are animals that were given the vehicle, and here's the animals that are given the antagonist. You don't have to squint. There's no effect there. Okay? I can tell you that when I got these data the first time, I said to Karen, run it again. Right? And she came back with similar data. I said, run it again. Right? There's something we're not understanding here. And in fact, it was really my failure to understand what we're looking for in the system. These are animals that are fed a chow diet, right? And are fed, right? Which means that they're on a vegan kind of diet, right? And don't have very high circulating fatty acids. So again, this is a situation in which there probably isn't very much normal PPAR gamma activity, so when we try to block it, it doesn't matter very much. So we had to go back to the drawing board and say, well, what are the two situations where you could imagine having where we'd have increased levels of the fatty acids that we believe would drive PPAR gamma activity, and that the animal would be hungry? Well, one of those is fasting, right? When you fast the animal, one of the things that happens is you're freeing up, lipolysis is going on, you're freeing up, and you're seeing elevated levels of fatty acids, okay? So now if you look at an animal that's fasted, what you see is that the animal goes from consuming 70 kilocalories to 100 kilocalories. Again, becomes hyperphagic. If we now block PPAR gamma receptors in the brain, what you see is that over 2/3 of that increase in food intake goes away, right? So again, here's a situation where the animal's hungry and increased fatty acids. Some of the increased food intake can be attributed to the increase in PPAR gamma activity. Now, you can imagine what the other situation is if you're following along, right? And that is what happens on a high-fat diet, right? So here are animals that are on chow, and you can see that the antagonist does very little. Here's an animal on a high-fat diet, and what you can see is that, in fact, now we can have this nice effect of the antagonist to reduce food intake. So again, I've showed you now two different situations under which you'd predict that you'd have high endogenous activity to this PPAR gamma system, and then by knocking down activity, in fact, you can see an effect. Okay? Now, let's get back to leptin. Remember, we started here with this problem about, well, what happens in terms of leptin resistance. So as I mentioned before, right, we know that leptin under normal conditions, you can raise the levels and cause a decrease in food intake and body weight. But when you feed the animal a high-fat diet, right, levels expand, the leptin levels themselves increase, but in fact the animal becomes less and less responsive to leptin. So the question is this an effect that we can reverse by blocking PPAR gamma in the central nervous system? Okay? So the experiment I'm going to show you is we are going to put all the animals on a high-fat diet, right? And we're going to give them leptin either under normal situations or when we block PPAR gamma in the brain. Okay? So here is the food intake in animals treated with a vehicle on a high-fat diet. Here's that effect, right, of leptin. Again, leptin resistance is very clear in this situation. But now, in fact, when we do it in the presence of the antagonist, right, you can see that you can restore normal leptin action to reduce food intake, and that also goes along with an ability to cause it to reduce food intake as well, or body weight as well. Okay? So, the point here is that at least part of what's going on in terms of understanding the molecular underpinnings has to do with the way that nutrients can interact directly with systems in the central nervous system that are important for processing that leptin signal. Okay? Now, interesting enough, and this is actually from the original paper from the University of Washington, where they showed that key components of the melanocortin system express the PPAR gamma receptor. Right? So these are neurons within the arcuate nucleus that either express AGRP or alpha MSH. So the idea that part of the way that PPAR gamma regulates food intake has to be dependent on the melanocortin system seemed a reasonable hypothesis. Even more reasonable after this paper that came from Sabrina Diano and Tomasz Horvath, in which they showed that PPAR gamma drugs could alter the activity of these key sets of neurons that regulate melanocortin signaling and regulate food intake and body weight. Okay. So what we went to is go back and now and say, well, what happens if we give PPAR gamma drugs in the presence of a melanocortin deficiency, right? So in fact, I have a very talented postdoctoral fellow in the lab by the name of Yoram Mule, and he had worked on these rats that are melanocortin 4 deficient. I won't bother trying to explain the model because it's not any different than the mouse model, except they're actually even fatter than what you see in the mouse, right? The point is, the question is whether PPAR gamma can still increase food intake in the absence of melanocortin signal. So here again, we pick a dose that has very little impact on the wild-type animals. This is peripheral rosiglitazone, right? But what you see here in the animals that are deficient of MC4, first of all, they're hyperphagic on their own, right? No surprise there. But they also have this enhanced response to PPAR gamma, right? And the exact opposite of what we had hypothesized. This turns out to be true for weight change as well after administration of TZD drugs. This is intake over a 12-day experiment, and you can see a small but significant effect of this dose to cause an increase in food intake that contributes to weight gain in wild-type animals. In the animals who are, in the animals who are don't have MC4 signal, you can see that they have an increased food intake. But you can also see that in response to rosiglitazone, their effect's even larger. If you look at their cumulative weight change, you can see that they gain even more weight, right? These are animals that are already obese. You give them the rosiglitazone drug, they even continue to overeat even more, right? And gain even more weight. Worse than that, right? If you look at glucose tolerance, so here are animals, the wild-type animals, again, in pretty good shape. You can't improve their glucose tolerance with rosiglitazone. Why? Because their glucose tolerance is already really, really good, right? But what you can see is that in the animals who are who don't have MC4, their glucose tolerance is worse, and in fact, that is improved by rosiglitazone. So my point here is that it isn't the case that the actions of PPAR gamma are dependent on the classic circuit that we've associated with the control of food intake and body weight in the melanocortin system. So we're going to have to continue to understand that this is revealing other parts of the circuitry that we haven't currently appreciated about how it is that it's having these potent effects that drive hyperphagia. Okay, I've got one last little vignette to tell you about. So what I've been trying to convince you is the idea that this CNS PPAR gamma system is important for regulating intake, particularly in situations in which you're on a high-fat diet, okay? Now, notice something, right? For those of you who are hypothalamic aficionados, I've showed you that there are PPAR gamma receptors in this area called the paraventricular nucleus of the hypothalamus. I bore you with that detail now because that is a place that regulates a number of aspects of our responses to stress, okay? Both endocrine, sympathetic, and behavioral responses to stress depend on activation of this PVN circuit in a number of profound ways. Okay? And so we hypothesized that, in fact, the PPAR gamma system was not just involved in regulating food intake and energy balance, but would also be involved in regulating the stress response. Right? And again, at the end here, I'll try to give you a reason why we thought that in terms of what the potential relationship is between psychological stressors and what happens when you're on a high-fat diet. So, again, like I said, the PVN is involved in the circuitry that controls glucocorticoid secretion, but also controls sympathetic activation and control the cardiovascular system. So here's the prediction, right? Is that we're going to have that increased PPAR gamma signaling will blunt the cardiovascular response to psychological stressors. So here's what the experiment was. So what we do is we implant these telemeters in the animals. And now we're going to, so we can monitor blood pressure and heart rate. And now we're going to give the animal 5-day treatment regimen with TZD drug. Okay? And then at the day 5, we put the animal in a restraint stress for 30 minutes, right? So we put them in a small space, and not surprising to anybody here, their heart rate goes up, right, when you put them in a small place, okay? Many of you probably experienced that on a plane today, okay? So what you see here is that this is mean arterial pressure. Again, blood pressure goes up, and that's not affected by whether you get vehicle or rosiglitazone. But if you look at heart rate is greatly reduced in the presence of rosiglitazone. That is, you have a reduced response to the stressor of being put into this tiny tube if we in fact have been treating you with rosiglitazone. Okay? So that's cardiovascular responses. How about looking at the endocrine responses? How about looking at the production of glucocorticoids? So again, similar kind of experiment where animals are going to get rosiglitazone. We're now going to put them in the restrainer, and we're going to take blood draws and look at the activation of the HPA axis as it reads out in the endocrine system. And again, no surprise to anybody here, when you put the animals in the restraint tube, right, for 30 minutes, their glucocorticoids go from very low to very high, right? And they keep going up even after you take the animal out until they start recovering 2 hours later. And what you see is that this is a very blunted response when you in fact treat the animal with rosiglitazone. Right? So if you look at brain responses to stress, right? So we can use a variety of markers to say what parts of the brain are being activated by stress. Again, when you look at this PVN circuit, right, in the presence of rosiglitazone, we stress the animal, we see less neuronal activation. And again, this is just a quantification of that picture. Right? So again, there's a number of ways in which rosiglitazone is actually reducing the response to stress. So even worse, if we actually antagonize PPAR gamma receptors, we make the response to stress worse, right, in looking at the endocrine system. So again, it's a endogenous, biologically relevant regulator of what goes on in terms of the HPA axis. So why do I bother you with this? I bother you with this because we spend a lot of time talking about leptin resistance and how it might contribute to increased weight and the maintenance of increased weight. The problem with the model is, how did we start overeating in the first place? Right? If we didn't ever choose to consume the high-fat diet, we never would have set up the activation of PPAR gamma that would have then turned it into leptin resistance that means that you become hyperphagic and maintain that higher weight. Right? So we often ignore what's going on at the initial event. Right? And so what some of this data leads me to hypothesize and speculate about is that what are some of the events in our lives that matter to why it is that things look worse for obesity than it did 30 years ago? Well, imagine that the world is more stressful than it was today compared to 30 years ago. Everybody says, no, that can't be true, right? Right? But the point here is that in that situation where we have more stress and more activation of that system that's there to accommodate stress, that the ability to drive ligands for PPAR gamma, right, would in turn turn down that stress system. Right? This is, to put it simply, a way to think about what comfort food is. Right? That high-fat diets can have a biological action, right, to suppress stressful responses in the key circuitry. And that, so part of the reason why high-fat diet availability then allows for greater consumption has to do with the fact that it makes you feel better, right? And then once it makes you feel better, the downside to that is it's also making you less responsive to the same hormones that would normally limit your increased intake and weight gain. Now, do I think that explains all of it? No, I don't want to pretend that I do, right? But the point is that if we step back a moment and stop thinking of the systems in terms of what we eat as only in their biochemical nature of how we process them into calories, but think of the foods that you eat as signaling molecules, right, that drive a variety of different receptors in different tissues, that we can build a much better understanding of how it is that what we eat contributes to disease, and how it is that we can build nutritional components that would actually limit those diseases. I'm not saying that we shouldn't be doing nutritional epidemiology, where we would go in, look at a bunch of people, ask them questions about who's eating what, and then looking how that relates to their disease state or their prevalence of disease. But this is a complementary bottom-up approach to it, to ask about how signaling from the foods that you eat matters to the potential biology that underlies the disease. And I think that those two things can complement each other, and I believe that this will help us understand both etiology and potential therapeutic strategy. I will stop there and point out the obvious, which is that I didn't do any of the work you just saw. Right? It was done by a variety of folks at the University of Cincinnati. Particularly, all the stress stuff was done in collaboration with Eve Ulrich-Lai and a number of folks that work with her. Mitch Lazar gave us the construct that we needed for some of this stuff. I told you about Yoram Mule, right, who did the work on the MC4 system. And then this is the larger group of folks. This is my longtime colleague and friend Steve Woods, who's an important part of what you've seen. This is Darlene Sandoval, who's an important collaborator of mine. And right there is Karen Ryan, who led much of the work on PPAR gamma in the brain. And with that, I will stop here and thank you for indulging me with this opportunity to come to you tonight and talk. And I'm happy to take a few questions if you have time. So thank you very much. So the question, if you couldn't hear it, was, are there any natural PPAR mutations in humans that where phenotypes might bear on this particular story? I don't know is the answer, right? We have a number of geneticists in the audience, however, right, who would be in much better position to try to answer that question than I. So again, what you would be looking for, the most obvious versions would be gain of function versions, right? And again, the relative potential for finding gain of function versus loss of function, again, the geneticist should comment rather than I. Those would be the ones that would stand out, because again, the loss of function. So here's the problem when you think about PPAR gamma, right? So PPAR gamma is a transcription factor that grabs both co-repressors and co-activators. So under its normal state, loss of function means that the co-repressors aren't there that actively inhibit gene transcription. So loss of function actually can increase PPAR gamma activity in terms of gene transcription, which is why we didn't do we didn't use the Krelok strategy for exactly that reason, because we were concerned that we wouldn't be able to see stuff, and so that complicates any sort of analysis here, because loss of function isn't loss of signaling inherently in the system. Yeah, so we see gain in all of the fat pads, right? But what we haven't done is something even more interesting, which is to try to look at, when we tickle the PPAR gamma system in the brain, how much you're changing adipocyte function, right? So we know that there's all of this regulation of adipose tissue that we can't explain just locally, that probably has to either come from endocrine factors or from CNS control. And the notion that PPAR gamma is acting both in the adipose tissue and in the brain simultaneously to direct adipose tissue function makes inherent sense to me, but we don't have any data to drive us one way or the other. Yeah? Yeah, so so the issue is about how do you figure out what are the right ligands, and are there strategies you could use in fed versus fasted situations to try to pull down what's interacting with PPAR gamma, right? Is that the right question? Yeah. So it's a really good question, and again, this gets beyond my my expertise very rapidly. People have been constantly looking for better PPAR gamma ligands endogenous ligands than we have today. And so every time there is one that somebody tosses out, we start getting really excited about trying to go either manipulate it in the diet or manipulate it in the brain in some way to try to drive the endpoint. The problem is the differentials between them are all relatively small. When you compare what a TZD drug can do to PPAR gamma versus any of the ligands that we have, it's not even close, right? Which implies that maybe there are, again, are novel versions of this that are floating around that we haven't appreciated yet that might interact with the PPAR gamma system. And you're proposing a way to begin thinking about and pulling them down. We haven't tried anything like that. It doesn't seem unreasonable to me. I don't know about how technically feasible it is, but the issue here is, but it's the right, if you're thinking of food as a source of a ligand rather than a source of a calorie, then trying to figure out which ligands out of the food matter would matter a lot in terms of understanding which diets contribute and which diets might help in terms of the long-term outcome. Okay. So the question is whether we've shown any of these effects with CNS infusions of fatty acids, and the answer is no. And again, the hard part about those experiments are complicated, right? How do you package them and deliver them into the CNS without doing damage is no trivial feat. Now, if you look through the literature, what you'll find is people talking about particularly different fatty acids having deleterious effects to turn on the system, right, in different ways. And so the question is whether that's PPAR gamma dependent or not, and the answer is we just don't know. We don't have the right tools to be able to do that rapidly. Okay. Thank you very much. I appreciate your attention.